Hello everyone, welcome to our science fair presentation. Today we will present a research conducted by scientists in Texas. This research shed light on a unique pathogenic pathway of Pseudomonas aeruginosa, a very common bacteria in hospital. Our presentation is divided to 4 parts. First, we will give a brief introduction of the research subject. Second, we will talk about the research process and results. Finally, we will share a conclusion of this research and our opinions on how this research can be further developed. Pseudomonas aeruginosa is a common encapsulated gram-negative rod-shaped bacteria. It can cause disease in plants and animals, including humans. It is considered opportunistic insofar as serious infection often occur during existing diseases or conditions, most notably cystic fibrosis and traumatic burns. Pyoverdines are fluorescent siderophores produced by P. aeruginosa to acquire iron. They need to do so because the solubility of biologically relevant iron compounds is exceedingly low, and is generally insufficient for the needs of most microorganisms. Like most siderophores, pyoverdine is synthesized and secreted into the environment when the microorganism that produces it detects that the intracellular iron concentration has fallen below a present threshold. They are important virulence factors and are required for pathogenesis in many biological models for infection. Because of its ability to chelates ferric iron, pyoverdine helps P. aeruginosa grow by acquiring iron and in turn damages host mitochondria, which are iron-rich organelles. In addition, it can also effectively contribute to the formation of biofilms that play a key role in the development of AMR. Nevertheless, by inhibiting the biosynthesis of pyoverdine, some small molecules can limit P. aeruginosa pathogenesis, providing a promising approach to tackle antimicrobial resistance in this microorganism. Scientists accordingly conducted research to find out the pathogenic mechanisms of pyoverdine and its importance in host-pathogen interactions.
In this experiment, worms were exposed to filtrate for several hours, and after that, pyoverdine fluorescence was assessed. Initially, pyoverdine fluorescence is quenched when binding with iron. Then, researchers use excess 8-hydroxyquinoline to strip iron from pyoverdine, restoring fluorescence. In assessing pyoverdine fluorescence, researchers observed a time-dependent increase in pyoverdine fluorescence within the host, as you can see in this figure. They subsequently confirmed that the majority of the pyoverdine within the host is not associated with circular membranes. Rather, it was located within the worms, particularly in the extraluminal tissue surrounding the pharynx and in the tail. Previous slides demonstrated that pyoverdine can get inside the host. However, the goal of pyoverdine is to provide the organism with ferric iron. Hence, in order to be useful for bacteria, it means not only to get inside of the host, but also to be able to leave the host with iron. To test whether the phenomena happen, a following experiment was set up. Worms that were loaded with pyoverdine was washed and frozen immediately or transferred to pyoverdine-free medium to permit the siderophore to exit the host. As you can see in this figure, free pyoverdine makes its way back out of the host. Within the first 2 hours, 40% of pyoverdine was lost, and the loss continued more gradually until at least 6 hours.
To find out the impact of pyoverdine translocation on the total iron pool in C. elegans, researchers used ICP-MS to measure the levels of iron in worms exposed to filtrate from pyoverdine deficient strain to filtrate from pyoverdine rich strain or media control. As you can see in these two figures, wild type filtrate is the one that is pyoverdine rich, and PVDA filtrate is the one that is pyridine deficient. And as you can see, worms treated with filtrate from wild-type bacteria rating 32.6% less iron than worms treated with filtrate from media control. In contrast, worms treated with filtrate from Pseudomonas aeruginosa PA14PVDA, the pyridine deficient strain, were almost indistinguishable from those treated with media. Since host mitochondria represent a rich intracellular pool of iron, it's conceivable that mitochondrial functions could be damaged when pyridine extract iron from host cells. Mitochondrial iron-sulfur proteins generate ATP via oxidative phosphorylation, which involves the oxidation of NADH to NAD+, making the ratio of these metabolites a sensitive indicator of mitochondrial function. So using a pyridox-based reporter, a protein that binds to NADH and fluoresces green upon binding it, researchers measured the cytosolic NADH to NAD+ ratio to assess mitochondrial function after exposure to pyridine-rich filtrate and saw substantial accumulation of NADH, the reduced form. The NADH to NAD+ ratio was significantly lower in worms treated with filtrate from PVDA, the pyridine deficient strain, iron-bound pyridine or media. So damaged or excess mitochondria are recycled via autophagic turnover in a process mediated by kinase PINK1. This protein is constitutively expressed and is rapidly imported into the matrix of healthy mitochondria, where it is destroyed by resident proteases. Mitochondrial damage compromises protein import, increasing PINK1 stability. This change can be observed in C. elegans by using a GFP tagged PINK1. A string carrying this GFP tagged PINK1 construct to pyridine-rich filtrate was exposed, and researchers observed strong stabilization of PINK1, which indicates that pyridine treatment was has compromised the mitochondrial membrane potential and protein import. So under normal physiological conditions, mitochondrial synthesis and turnover are carefully balanced to maintain cellular homeostasis. Researchers hypothesized that pyridine exposure is likely to disturb this balance, tilting it toward mitochondrial turnover. To test this, the ratio of mitochondrial to nuclear genome counts in worms treated with either pyridine-rich filtrate or filtrate from a pyridine-deficient control was measured. They obtained the ratio by measuring the amount of nuclear DNA and the amount of mitochondrial DNA. If autophagic recycling of mitochondria is activated, there will be less mitochondria and the ratio would be reduced. Pyridine treatment would reduce this ratio, indicating that damaged mitochondria are being recycled. Last, one of the most important functions of mitochondria is making ATP. Pyridine exposure significantly decreased cellular ATP content, suggesting that pyridine-mediated mitochondrial damage also compromises ATP production, which is a vital metabolic process to fuel cells.
To see how the hosts defend in response to pyridine invasion, researchers performed the following experiments. First, microarray analysis was used to compare transcriptional profiles of worms exposed to purified pyoverdine media or untreated. Afterward, upregulated genes from pyoverdine exposure were compared to genes upregulated after exposure to exotoxin A, a translational inhibitor produced by P. aeruginosa, or genes upregulated during infection with P. aeruginosa on agar plates, where pyoverdine isn't produced. In this way, they can tell if the hosts respond by identifying the pathogen as something produced by P. aeruginosa or identifying the specific damage caused by pyoverdine. While 68 genes were upregulated by both tox A and P. aeruginosa infection,
Almost half of the genes upregulated by tox-A. Only 16 and 9 genes were in common between pyoverdine and P. aeruginosa infection on agar or tox-A, respectively. While statistically significant, these shared genes likely comprise a relatively nonspecific generic stress response. This suggests that the host can differentiate between what pathogen is doing on the agar plates and what pyoverdine is doing to worms in liquid. This conclusion was bolstered by QRT-PCR analysis of 12 genes selected on the basis of significant upregulation after pyoverdine exposure and to represent several classes of genes. Expression of these 12 genes was also assayed in worms exposed to phenanthrene, which were compared to worms exposed to DMSO solvent. Pyoverdine and phenanthrene have different molecular patterns, but since they both are iron chelators, they cause similar damage to the host. In each case, the same expression pattern was observed, suggesting that the host is responding to perception of damage that is the same after each treatment. There are 3 major conclusions for this research. First, pyoverdine can translocate into the host, acquire iron, leave the host, and bring iron back to bacteria. Second, removal of iron results in mitochondrial damage and subsequent host death. Third, worms defend by activating specific immune pathways that is likely related to the damage inflicted. Finally, the potential future directions of this research project. First and foremost, given that the mechanism by which pyoverdine gains access to and removes ferric iron remains unknown, researchers can delve deeper into this area. After learning about the mechanism, scientists can efficiently design new methods to limit pyovandine's access to iron. Second, they can conduct research on whether siderophores can enter cells and how siderophores liberate iron from host iron sequestering proteins. Third, scientists can develop new molecules that can find ways to stop pyoverdine from translocating into the host, preventing iron acquisition altogether.
That's our presentation, and thank
Thank
you
you
for
for watching.
Thank you for watching.